I'd like to thank well, several people, but specifically the students of ours that have returned for this event. I'm pretty sure I speak for both Mr. Maxwell and myself when I say that one of the greatest honors we get to receive is when our students return to see us. And so Maggie Morash, Michaela Ennis back there, he's trying to hide. Brooke Conti, thank you for coming back. And I don't know if you guys heard, but Maggie Morash, would you mind standing for just a second, Maggie?
I just read about it.
She's a Gold Medal Scholarship at Rutgers, and we're very, very proud of her. I love the fact that she's so embarrassed by the publicity that she's getting. So this is just my... excuse me. You know, few people understand the level of research that we're trying to do here. We're doing nothing like modeling research here. What we're trying to do is the actual, very real research. We want to be able to grow cancer cells here. We want to be able to do brain dissections here. And in order to do that, the infrastructure that we require is substantial. There's little things that you don't think of when you need to design a lab that is capable of housing a project like that. Something as simple as dirty air can destroy projects for years, as it actually has for us. We've been trying to do a technique called cell culture here for about 2 years. And we can only get it to work for about a week because it's difficult to clean the air in our labs. It needs to be far cleaner in that area than it can be out here. And to that end, we're doing a lab redesign that's going to cost us around $1.4 million. It's going to completely revamp the science wing, and it's going to allow us to do it's going to allow us to expand an already impressive research structure. We are about $600,000 into our goal right now, so we do have a little bit of a way to go. And if you can help us in any way, whether that be financially, with your expertise, or with your time, we would love your help. Let me direct you to David Mayer, who's standing up here. If you can get in contact with him, if you can offer any of those things, we'd really, really appreciate it. So let me tell you about David Maxwell, who's about to speak. Now, Dave is about probably one of the smartest men I've ever met in my entire life. I mean that in all sincerity. And I don't think you guys understand how good of a person he is. When I was interviewing here, he was my contact. And when I called him, he was on a bike ride. He stopped his bike ride to have that phone conversation with me so that I would have my interview here and be comfortable. But what you don't know, and what I didn't know until years afterwards, is that he thought I was going to replace him. So this guy took the time in the middle of his workout to help a guy, a new person out, that he was fairly certain was going to replace him. And thankfully, I have not. I get to work alongside of him. One of the reasons I think Mr. Maxwell is so intelligent is because of the way he looks at issues. He never looks at things as though they're one-sided. He always appreciates both sides of an issue. And it's one of the reasons here at Pingree we're able to take on such controversial issues. Because we don't necessarily propagate a certain idea as much as we encourage a certain discussion. Now, the genetically modified organism topic is one that is hotly debated right now. It is huge in Europe. The fear associated with it is great, and the knowledge that we need to combat that fear is also great. So, it is with great pleasure and an honor to introduce my colleague and one of my best friends, David Maxwell.
and thanks for putting this whole thing together. He's the architect of this whole thing. So I'd like to point out also that this day is not about me. This day is about the students. And I'm giving a presentation, but the kids have done some really, really amazing things. I'd like to thank them for all their hard work. So, without further ado, I'd like to talk to you about a topic that I'm pretty passionate about. You all know, I'm sure, are familiar with these species that are endangered. The bald eagle, of course, is an iconic American symbol and has recently been downgraded off the list, right? That's a wonderful, wonderful thing. And wouldn't it be cool if we could do that for all of these? For each one of these up here, that's a beautiful poster child for the topic of extinction. There are lots of organisms that are not. My nephew is not one of them. So, one of the tools that we have, the question is, should be used if people are worried about it, is genetic engineering. And we're going to talk about that a little bit today. As Mr. D said, if you look in the popular media, this is kind of the image that you get of what genetic engineering is. That you're going to have, snail genes in your broccoli, both of which frankly are disgusting. Trout genes in tomatoes. And remember that a gene is just a piece of information, and most of the information that makes up organisms is the same. Okay? So, anyway, there are some legitimate concerns that we need to think about when we're going to do something like take a gene from one organism, put it in something that's completely unrelated to it. And these are some of those concerns. We'll come back to each of these later. So this one was supposed to be moved, but since it hasn't been moved to later, we'll talk about it now. What you see here, beautiful, pristine forest, right? This is the forest as it is out there right now. But what you probably don't know is that this is actually a very degraded environment. 200 years ago, the tree species that were out there are very different from what they are today. So imagine 1/4 of the trees that are out there being replaced with something different. Okay? We'll come back to that. I'm not going to get into the details of that, but we'll come back to it right now. Okay. American chestnut is what those trees used to be. We don't know this anymore because, as far as I'm concerned, I was born in the 70s and this happened in the 1900s. But the chestnut is a iconic part of our heritage. It's like the American bald eagle for the forest. Okay? And this is what happened to American chestnut in a very short period of time. It went from huge forests to that in about 30 years. The fungus was introduced into Newark on some wood, and by the 1950s, it had decimated the whole forest finally. What it does is it enters in a wound, and then it grows in rings until it kills the whole tree. Once it's cut off the stem from the leaves can no longer put sugar and send it down to the roots can't send water up to the leaves, the tree dies. There's a toxin, which I'm going to refer to this toxin just as a toxin, but it's called oxalate, and that's what kills the cells that the tree eats. Now, there's been an effort for the past 30 years to breed for resistance using traditional breeding methods. And each one of these steps here takes 5 or 6 years. Okay, so you start with an American chestnut and a Chinese chestnut, and what you get out at the bottom is something that's mostly American chestnut, but also has the resistance of Chinese, because Chinese chestnut is resistant. You can do this faster. Okay? So in 12 to 18 months, you can get a resistant chestnut tree. And the way you do that is you take a gene, which is basically an antidote gene to that toxin, and you put it in your tree. And this has been done for apple, and you can see that the one that has no antidote gene, the fungus grows faster than it does in the one without the antidote gene. Okay? So fungus produces toxin, plant now produces an antidote, and it can resist the fungus. Any questions so far? When there's silence, I always wonder whether I'm actually getting it or whether you just are totally lost. Okay, so how the heck do you do this? How do you put that gene into a plant? Well, the gene that I showed you comes from wheat. So we know that we eat wheat, and wheat is not toxic to us unless, it's gluten, but this is not a gluten gene. So what we do is we take that gene and we put it into Agrobacterium tumefaciens, which is a natural genetic engineer. What this bacteria does normally in its life is it sticks DNA into plant cells and hijacks the plant cell to make a sugar that only those bacteria can eat. What we do instead is we take our antidote gene and we put it in here, and then we let the bacteria put our antidote gene into the plant cell. And then from there, well, this is a rare event. Okay? So the problem is, not every plant cell gets the hunk of DNA at the end. Okay? So what we need to do is figure out which of those cells actually have that information, that gene, that hunk of DNA. And so what I'd like you to do now is stand up and look around and see if there's a piece of paper associated with the gene. So go ahead and stand up and look around. So what we're going to do is you represent cells, your plant cells. We're mixing agrobacterium with you. And if you have one of those pieces of paper, stay standing, because what I'm going to do is I'm going to release a toxin into the environment. And anybody without a gas mask is not going to be alive. So if you have a gas mask, if you're one of these cells, you can sit down now. Thank you very much. If you have a gas mask, you're one of these cells that got that piece of DNA. And then when those cells are plated in agar media, every cell that lives got that hunk of DNA. And so all you have to do then is, you do a little bit of manipulation of hormones, and you can grow that single cell into a mature tree that has your piece of DNA that has the antidote gene on it. So the gene that it has an antidote gene that we want to put in there, but it also has the gas mask gene. Okay. So to get the cells the right way, we have to put both the antidote gene and the gas mask gene. Does that make sense? Because that's important. One of the concerns has to do with that particular piece of information. So, what Dr. Zhang did was, he's the guy who did this research, and what he did is, to figure out if something's resistant, you have to have a bioassay. And so, you can take a leaf off of a tree, make a little wound here, and then put the fungus on there, and then see how far it grows. This is Chinese chestnut, which is resistant, and then this is American chestnut. You can see the fungus grew a lot farther. And so, if we test our trees, we see here's how long the necrotic dead spot is. And this is your American chestnut, which is, remember, your susceptible variety. This is Chinese, and this is your gold standard for resistance. And here are two of the transgenic trees that Dr. Zhang made. So not only do we get resistance in 12, but we also get better resistance. Mark, you had a question, or am I going long? Okay. So compare the two methods, traditional breeding, 25 years, and you're adding about 1,800 extra genes to the genome, which we don't even know what those 1,800 genes are. Compared to the genetic engineering method, a lot faster, and you're only adding 2 genes. And not only that, but we know exactly what those 2 genes do. Oh, and the resistance level path. So for my money, that's a way to go. But we know it's not an allergen because we eat it in wheat. Non-target organisms is something that people are actually looking into right now. Does it affect anything else in the environment? Because we don't want to mess up the environment by putting this. One worry is that extra piece of DNA, that is gas mask DNA, could get into other organisms. That's a legitimate concern. You have to think about that. And the final thing that most people think about is biodiversity. And what we're really doing here is replacing biodiversity, because American chestnut was that keystone species in the forest. Putting it back gets us more biodiversity. Okay, so there's obviously a balance that you have to strike. And I'd like to ask the question, if we don't use every tool at our disposal, are we actually being good stewards of those? Obviously, to go back to the big picture, there are other uses for genetic engineering. You can, if you have a plant that's resistant, you don't have to use as much pesticide. You can get more food in a shorter amount of time. You can cure things like vitamin A deficiency, which leads to blindness. And you can grow plants that don't need as much water. And so, since we're going on to where we're going to have, 9 billion people in a short period of time here, if you have the tools to feed the world, would you use it? So, I'd like to thank these people, Amanda and Sydney. Put them through heck time by listening to this presentation. They did a very good job of making it not stick. Thank you very much.
I'd like to also point out that Pingree Science and Pingree Research is, depending on how you look at it, one of the best programs in New Jersey, and according to our Siemens and Intel wins, one of the best in the nation. And Mr. Maxwell is now the head of the Pingree Science Department. So. He's got big ideas, and he's already implemented many of them, and we look for what he's going to do in the years to come. There are many awesome things to see out there right now. In the next half an hour, you can place a cardiac stent, you can mimic a brain surgery, you can isolate DNA, you can listen to a lecture on quantum physics and Buddhism. Make sure you make the next half an hour count. It's all you've got, because we will be seeing you back here at about 12:30 for Gender Neuroscience with Claire Putman. See you guys in half an hour.
That gets you very far. Nice to meet you.